Those of you going through a calorie strict diet in order to lose fat, you might have already heard the term autophagy. But then if you have not, you are going to learn about it right now, at least from the pathology point of view. So if you break the term into two, you get auto and phagy, where auto means self and phagy means eating. So autophagy is such a phenomenon of the cell where the cell eats itself. And since the cell is eating its own components, we also call it cell cannibalism. Now the main question is, why is it that a cell would resort to eating its own components? And the answer is for survival. So autophagy is indeed a survival mechanism. It's a very brief explanation to how autophagy takes place. This right here is a normal cell. It has your typical endoplasmic reticulum, ribosomes, mitochondria, nucleus, and most importantly, this yellow color organelle that is lysosome. Let me label it because it is important for the process. In yellow, filled with digestive enzyme, you have lysosome. So in autophagy, first the cell receives trigger. Now what these triggers are, I'll explain later, but for now, the cell receives trigger for the cell to go through the process of autophagy. As soon as the cell receives trigger, it starts eating its own components. That is, it starts performing cell cannibalism. And it does so with the help of organelle that I have just labeled, that is lysosome. It is inside the lysosome where endoplasmic reticulum, mitochondria, and ribosomes are sent. Inside the lysosome, with the help of lysosomal enzyme, these organelles they get digested. Once digested and broken down into their components, these components are then recycled by the cell so that they can be reused. This is how the survival mechanism takes place. So when is it that a cell resorts to showing autophagy? Let me give you a very simple example. When you are hungry, it is your cell that is hungry. When you are starving, it is your cell that is starving. When you eat food, it is your cell that eventually receives that food. When you are energized, it is your cell that is able to produce energy for growth. Now let's say this cell right here is starving because it is not receiving any nutrient. And in lack of nutrient, the cell has no energy. When the cell has no energy, it is unable to show growth. Eventually, the cell will become old and then die. But then do we want our cells to die? No. And the cell also doesn't want itself to die. So it will resort to eating its own components. The cell in wars of death, it will start eating its own endoplasmic reticulum, its own mitochondria, so that it can break down the endoplasmic reticulum, break down the mitochondria, and then produce energy for survival. So when the cell does not get food from outside, it will resort to eating its own components so that it can produce energy by breaking down its own cell organelles. And this is just one of the many reasons why autophagy takes place. So let's learn about those many reasons why autophagy takes place. Now the beautiful thing is autophagy takes place both in physiological condition and in a diseased or pathological condition. Starting off with physiological autophagy. Number one, aging. When you grow older, when an individual grows older, it's the cell within their body that is growing older. And the cell within the body contains cell organelles that are growing older, that are becoming aged. Now, what the cell does is that it eats, with the help of lysosome, it eats the old endoplasmic reticulum, the old mitochondria. They get eaten up by the lysosome and broken down within the lysosome and then recycled so that new endoplasmic reticulum, new mitochondria can be made and the cell can rejuvenate itself. So autophagy in a way is stopping aging. not literally stopping aging, but at least promoting longevity, pushing aging even further away. Second is exercise. When you exercise, you require more energy. So basically your cell, they require more energy, and to produce that supplement or extra energy, it will resort to eating its own organelles so that it can break it down and then produce the required energy. Number 3, erythropoiesis. During erythropoiesis, you have your reticulocyte, which is immature RBC, which is converted to erythrocyte, which is mature RBC. The reticulocyte contains cell organelles. Erythrocyte does not contain cell organelle. Where does the cell organelle go? They get eaten up by the process of autophagy. Number 4, fasting. Same thing. When you are fasting, you are starving yourself under a controlled situation. So your cell lacks energy and it will resort to eating its own component. And come to think about it, if the cell is eating its own component during fasting, isn't it also eating old mitochondria and endoplasmic reticulum in order to replace it with new ones? So could it be that fasting helps with pushing aging further away or support anti-aging? Maybe we will learn about that under the topic of aging itself. But for now, pathologic autophagy. Number 1, starvation. So starvation, same thing. The cell has no energy, no food. It will resort to eating itself. Number 2, cancer. The cancer cell, they take up all the nutrition from the body because they divide a lot. But then what about the other cells? No, they don't have any nutrition. The other cells now have to resort to autophagy for survival. Number 3, neurodegenerative disease. During neurodegenerative disease like Alzheimer's, Parkinson's, the neuron, they get filled or accumulated with degenerated or misfolded protein. The neurons, they are filled with degenerated or misfolded protein. Autophagy helps in eating and degrading and getting rid of those misfolded protein. Number 4, infection. So during autophagy, it is not just the protein, ribosomes, endoplasmic reticulum, mitochondria that get eaten up by the lysosome and digested. Even the infections that enter the cell, infection that enters the cell are eaten up by the lysosome and then degraded by the lysosome, like Mycobacterium, Shigella, HSV1. Okay. Now, types of autophagy. There are altogether 4 types of autophagy. Macroautophagy, microautophagy, chaperone mediated autophagy, and mitophagy. Again, macroautophagy, microautophagy, chaperone mediated autophagy, and mitophagy. Very simple, very simple. See, macroautophagy, as the name says, it has macro. That means the process is going to be longer than others. Plus, in macroautophagy, large amount of cellular components are being eaten at once. Mitochondria, endoplasmic reticulum, ribosomes, they are getting eaten up at once. Microautophagy, as the name says, micro, it's a shorter process. Chaperone mediated autophagy has the involvement of a very special protein, that is the chaperone protein. And mitophagy is nothing but macroautophagy. But in macroautophagy, I said what? You have mitochondria, endoplasmic reticulum, ribosomes getting eaten up at once. In mitophagy, the same process of macroautophagy takes place, but only for mitochondria. Only mitochondria. Okay. And to make things very simple, I'm going to show all 4 processes with the help of diagram in a single cell. So here's a single cell. Let's look at all 4 processes. Process number 1, macroautophagy. Macro, it's going to be a longer process and lots of things are getting eaten up at once. So you have mitochondria, endoplasmic reticulum, ribosomes. See, there's a bacteria right here. They are getting eaten up at once. Now, it has 4 major steps. Step number 1. In macroautophagy, there is formation of a plate. This plate it is derived from plasma membrane and the membrane of membrane bound organelle like mitochondria and endoplasmic reticulum, because this plate is also a membrane on its own. So this membrane is obtained from other membranes, the plasma membrane and the membrane of mitochondria and endoplasmic reticulum, and we call this membrane phagophore. Phagophore. So first step is phagophore formation. Second step, the phagophore or the plate on which you have all the food to be eaten, the plate grows longer. So there is elongation of phagophore. Elongation of phagophore. Finally, the phagophore gets completed to form a autophagosome. Autophagosome. You know what a phagosome is, right? See, you have a cell right here. This cell engulfs a foreign body inside. Okay, it engulfs a foreign body inside. This is called a phagosome. But in this process, is the cell engulfing a foreign body? No, it is not. It is engulfing part of its own cell. That is what self phagosome. Phagosome of its own content. So autophagosome. Remember, a very important protein name, remember, which is involved in both the elongation of the phagophore and the formation of autophagosome, and that is light chain 3 protein. Light chain 3 protein. Okay. Now, the autophagosome, it goes and combines with the lysosome to form autophagolysosome. Simple. One last revision. So what do we have? You have macroautophagy. In macroautophagy, you have mitochondria, endoplasmic reticulum, ribosomes getting eaten. But then first, all these components, they are kept on a plate. This plate is called a phagophore. The plate then elongates. The elongation of a phagophore is mediated by which protein? Light chain 3. The plate eventually closes and forms a autophagosome. The closure of the plate in order to form autophagosome is also mediated by which protein? Light chain 3. The autophagosome, with all the cellular components to be digested, eventually fuses with lysosome to form what? Autophagolysosome. Now the components which I'm showing with my nose inside here is sent into the lysosome, and inside the lysosome, the lysosomal enzymes that digest all the organelles. Okay, so that was macroautophagy. In macroautophagy, you have phagophore and autophagosome formation. Number 2, what is the marker? The spatial marker, it is light chain 3. Right. Microautophagy. As the name says micro, that means it is a much shorter process. Let's say right here we have old endoplasmic reticulum. This old endoplasmic reticulum in microautophagy is directly engulfed by lysosome by the process of endocytosis. This old endoplasmic reticulum goes here, and then from there it is directly engulfed within. Just like that. See, you have the old endoplasmic reticulum, which is endocytosed within the lysosome directly. So in microautophagy, there is direct lysosomal endocytosis. Okay. Next, chaperone mediated autophagy. Okay, within the cell, what you have here is a misfolded protein. Now within your cell, there are special proteins called chaperone proteins. These chaperone proteins, again, these special chaperone proteins, they bind with the substrate present on the misfolded protein. The chaperone protein, they bind on the substrate present on the misfolded protein. Now the function of this chaperone protein is to recover or correct the misfolded protein. Again, what is this? This is your chaperone protein. The chaperone protein goes and bind to the substrate of the misfolded protein. What does it try to do? It tries to correct the misfolded protein. But if it is unable to correct the misfolded protein, what will happen? The misfolded protein will keep on accumulating with the cell, and the cell will grow older and then die. So in order to prevent that from happening, the chaperone will now mediate the process of autophagy. Again, there is misfolded protein inside the cell. The chaperone will go and bind with the substrate of the misfolded protein. The chaperone will try to correct the misfolded protein. But if it is unable to correct the misfolded protein, in order to get rid of the misfolded protein, it will mediate the process of autophagy. How? It will escort this chaperone protein. It will escort the misfolded protein like this. See, it will escort it to the lysosome. Within the lysosome, there is a gate that is present. Through this gate, the misfolded protein enters inside. Shoot. Okay. And inside the lysosome, the misfolded protein gets degraded by the lysosomal enzyme. Now the question is, what is the name of this gate? See, this gate right here is a protein, and this protein is present at the membrane of the lysosome. So we call it lysosome associated membrane protein, type 2A. Simple. Lysosome associated membrane protein. Okay, that's it. Chaperone mediated autophagy. It is mediated with the help of chaperone protein. Some famous chaperone protein are HSC, HSP. HSP stands for heat sought protein. Second, the gateway for entrance into the lysosome is the lysosome associated membrane protein 2A. Final, mitophagy. Mitophagy is nothing but mitochondria specific macro autophagy. So all the process that I showed you in macro autophagy, that is the formation of a phagophore, elongation of phagophore, formation of autophagosome, formation of autophagolysosome, and then the digestion of the organelles. The same thing takes place. The only difference is, instead of all these organelles, you only have mitochondria. That is mitophagy. Okay? Alright, that's it. With that, we are done with autophagy. But then few clinical correlations related to it. Autophagy is mediated by a gene, which is autophagy gene. Take AT from autophagy and G from gene. The most important autophagy gene is autophagy gene 1. There are mutations that, if take place in certain autophagy gene, can result in certain diseases. For example, mutation in autophagy gene 5 can result in tuberculosis. Mutation in autophagy gene 16L1 can result in inflammatory bowel diseases like Crohn's disease and ulcerative colitis. Another, mTOR pathway, really important. mTOR is inversely proportional to autophagy. What do I mean by that? Within your cell, there are various nutrient sensing signaling pathways. What do I mean? So see, your cell, it requires nutrition for growth and development, and your cell also has certain ways for it to sense the presence or absence of nutrition. When it senses the presence of nutrition, the cell will go through growth and development. When it senses the absence of nutrition, it will not go through growth. So one of the ways it senses it is by a signaling pathway called mTOR. Now within the cell, if mTOR is activated, autophagy does not take place. But within the cell, if mTOR is not activated, autophagy will take place. Okay, let me explain you once again with the help of this diagram. This right here is a cell. Okay, the same cell in condition number one is well fed. That means it has lots of nutrition. So the cell is going to detect the presence of nutrition with the help of mTOR. And mTOR is going to inhibit autophagy. That is cell eating itself. Why would cell eat itself when it has plenty of food already? Condition 2. In condition 2, the cell is starving. There is starvation. So basically, this will inhibit the signaling pathway mTOR. And when mTOR is inhibited, autophagy is active. Because now, in absence of food, nutrition, and energy, the cell will eat itself in order to produce the energy required for its survival. There is a drug by the name of rapamycin. Rapamycin, it inhibits mTOR. When mTOR is inhibited, autophagy is activated. So I'll talk about rapamycin once again on the topic of aging, but for now just understand that rapamycin, by inhibiting mTOR, is supporting or activating autophagy, that is cell eating its own component, and in turn preventing the process of aging.